During IVF treatment, it's typical for several embryos to be produced, and yet they can't all produce a baby. It turns out that one of the main reasons why so many IVF embryos fail to produce a child is that they have chromosome abnormalities in their cells. Now, chromosomes are small rod-like structures that exist in virtually every cell of our bodies. We have 46 in each cell, 23 from the mother and 23 from the father. But what we find in IVF embryos is often they have maybe one chromosome too many or one too few. And the effect of this is to mean that the embryo is unable to produce a child. Most often it won't implant, or it might miscarry very early in pregnancy, or occasionally produce a child with a chromosome abnormality like Down syndrome. For this reason, we've developed a technology that allows us to look at the number of chromosomes in the cells of embryos produced during an IVF treatment. The idea being that we can find those embryos that have the correct number of chromosomes, those embryos that have a chance of making a baby, and make sure that those are the ones given the top priority for transfer to the uterus. By avoiding the transfer of the abnormal embryos, things like miscarriages or failed implantations made much less common. Similarly, the risk of problems like Down syndrome is greatly reduced after this kind of approach. Now, Juno Genetics has developed a specific test for this called PGT-A Seq. We believe that this is the most accurate way of looking at chromosome copy number in embryos produced via IVF that's available anywhere in the world at the moment. Because of this very high accuracy, the risk that we would accidentally discard an embryo that might be viable is extremely low, and that's been proven in clinical studies, which is not the case for most other tests that look at chromosomes in embryos. So by applying this technology, we can think about getting a faster time to pregnancy, less time wasted on transfers that will not produce a child. That's going to reduce costs, reduce stress, and just streamline the whole IVF procedure. Less risks of miscarriages, which of course can be very traumatic, especially for a couple who desperately want to have a pregnancy. Less risks of severe abnormalities in children born. Another advantage is that if there are embryos frozen, we will know whether they have a good chance of making a baby or no chance at all. And so this means that sometimes it's not necessary to maintain the freezing of these embryos. So PGT-A Seq really is a unique procedure. It uses the most accurate and state-of-the-art technologies available to us at the time. And we're really excited at Juno Genetics to be able to introduce this and offer it to the clinics that are working with us.